Right, so good afternoon, everybody. My name is Alexandra Albu. I am a PhD student at UBB, and today I will be presenting a deep learning approach that we've been working on, which is a method based on autoencoders for predicting protein-protein interactions. Um, I will start a bit with a couple of words about autoencoders. They are one of the most widely used and well-studied unsupervised representation learning methods, which are actually neural networks formed of two components: an encoder, which is a function that maps the input data to an intermediate representation, a high-level representation of the inputs, and the decoder, which is used to reconstruct back the input using this intermediate representation. And this model is unsupervised learning algorithm because it does not require any type of annotations about the data, just the data itself. Now, while studying the properties of the latent representations that are obtained in this way is an interesting question on its own. There has been interest in seeing or studying the way in which autoencoders can be used in downstream tasks, such as classification or regression. And there have been mainly three directions for this. So the first one is that of using autoencoders as feature extractors, in the sense that in this setting, we have an autoencoder which we train on in an unsupervised training stage at the beginning. Then we can drop the decoder and simply use the first part, the encoder, to obtain these high-level representations that are further used in order to train any type of machine learning classifier instead of the inputs. So in this way, we have a frozen encoder that simply obtains a different representation for the inputs that can be easier to process, or because it would be low dimensional, it can it can help the learning process of the machine learning classifier. However, in this way, the challenge is that because the two stages are performed independently, it can happen that the features learned in the reconstruction phase are not necessarily useful for classification, or useful features and less useful one can be entangled, and it may be difficult for the machine learning algorithm to classify and learn this task. A second direction aims to improve upon the first, in the sense that the first stage is identical. We have an unsupervised pre-training of the autoencoder, but further we don't keep the encoder frozen, but we fine-tune its weights by adding a neural network classifier on top of the encoding layer and train the entire network. And in this way, we can adapt the encoder to better match the classification task. A third direction I will be talking here about is rather used in anomaly detection applications, in which we have a majority class, and we want to be able to detect points that are not sampled from the same distribution as the training points. So we can do this by training an autoencoder on this majority class that we have, and further detecting outliers by seeing which of the points give a high reconstruction error, so meaning that they are not recognized by the autoencoder as belonging to the same class as the one that it had seen during training. Coming back a bit to the application that I am presenting, why is protein-protein interaction prediction important? It is a subfield of computational protein data analysis, which is relevant because proteins play important roles in all biological processes inside living organisms. And a central problem in computational biology is being able to determine protein functions starting from the protein sequences. So only from the sequence itself, being able to determine a function that that protein will have. protein-protein interaction prediction is linked to this problem because the majority of proteins perform their roles not independently, but in complexes formed of multiple such interactions. These interactions can be determined experimentally. However, this procedure is expensive and also can be prone to false positives. So there has been an interest in being able to design computational approaches that can overcome these limitations and can reliably detect if a pair of protein sequences interact or not. Since in training these machine learning models, we actually use the experimentally determined data. The labels of these interactions can be noisy, which is a challenge in training properly these types of models. And another difficulty arises from the fact that we have a much smaller number of known interactions compared to all the possible pairs that are not interacting, or possible pairs of proteins that are not interacting. So this creates a highly imbalanced data set. Nevertheless, there has been quite a body of work on using machine learning methods for protein-protein interaction prediction. So starting from supervector machines and random forests up to ensembles of such classifiers or ensembles of neural network classifiers. Another quite widely studied idea is that of trying to design, especially when working with neural networks, in trying to design some architectures that are specialized for paired input data, because in our setting, our samples are actually a pair of two other items of two proteins. So from this perspective, Siamese architectures have been used, which are actually neural networks that have a shared network, so a set of weights that is shared between the two proteins in the pair, and in this way can capture common characteristics of the two proteins and can help in better modeling these relationships between the proteins. These types of architectures have been studied in various flavors, from plain convolutional architectures up to convolutional and recurrent architectures, all types of such models. Autoencoders have also been studied in the context of protein-protein interaction prediction as feature extractors. So this would be the first direction that I talked about earlier, and in which, in this case, a supervector machine was used as the final classifier, but also using the second idea that I mentioned, that of fine-tuning. So having an autoencoder as pre-training stage and then fine-tuning the weights using a neural network classifier. Third idea that has been investigated here is that of using autoencoders on graphs, because the protein interactome can be seen as a graph in which the nodes are the proteins and the edges are the links between the interactions between the proteins. And such a model is trained to learn embeddings for the nodes using all their neighbors in the graph, and in this way can learn more expressive representations. And it is a model that is specialized for learning on graphs and can be used for link prediction on graphs. The approach that will be further presented was published last year and is more closely related to the third direction I talked about, namely that of anomaly detection. However, in this setting, we have two classes. So we will have two autoencoders, one for each class, and each of the models is trained to reconstruct its specific class. During testing, we are able to classify a pair of proteins by evaluating which of the two autoencoders is able to better reconstruct that testing data point, meaning it's able to recognize it as belonging to that particular class. This is a visual representation of the approach. So as I mentioned, we have a set of protein interactions, which are pairs of proteins, which we can split into positive and negative ones. And we further train a dedicated autoencoder on each of the two classes. And during the evaluation phase, we can pass the testing sample through the two models, and we can compute the probability of belonging to one class or the other using the losses obtained by the two models on that testing sample. And now, since, as I mentioned, we have pairs of proteins, we thought about designing architectures that are more specialized for this type of input. Namely, we proposed two Siamese architectures, two Siamese autoencoder architectures, which formed in the following way. So first of all, we thought of having a Siamese encoder, only the encoder, which is actually a neural network that is used to encode both of the two proteins. And after obtaining these intermediate representations for the proteins, these are fused and given as input to a decoder, which reconstructs back the concatenation of the two proteins features. So in this way, the decoder is not a Siamese one, but is a merged or mixed one. The second idea was to have both an encoder and both the encoder and the decoder as the Siamese architectures. But if we left the architecture just like that, we would end up reconstructing independently the two proteins in the pair, which is not something that we would want, because the same proteins can appear both in the positive and in the negative classes, but paired with different proteins. So we wanted to have a type of reconstruction that that reconstructs actually the pair, not the individual proteins independently. So the way in which we opted to do this was by creating an additional representation, which is a fusion of the representations of the two proteins, and then give this pair representation to both decoders in order to reconstruct the two proteins. Actually, it's the same encoder, but the representations are different. We compared these two Siamese architectures with the baseline architecture, in which we simply concatenated from the beginning the features belonging to the two proteins in the pair, and we trained the model to reconstruct these concatenated features. And this type of approach can be considered a sort of early fusion, in the sense that we don't process in any way using an additional neural network these protein features, but we simply have a simple operation here to fuse them and use the model to simply reconstruct this representation. The way in which we represented protein data was using two widely used representations from the literature that aim to capture both physical chemical properties of the amino acids in the sequence, but also to capture some sense of the sequential nature of the sequences by computing some correlations between amino acids situated at some distance in the protein sequence. And in order to get the best of both worlds, we combine these two representations by a simple concatenation, and in this way we obtain a protein representation. Our model was evaluated in a similar way to other approaches from the literature, in the sense that we use a cross-validation procedure, and we use the same number of folds as the related work on that specific data set. And we have evaluated the model using the classical classification metrics, evaluation metrics. The four datasets that we have used are public ones. The first one is a human dataset, while the other three datasets are related. They are multi-species dataset that were created by merging data interaction data belonging to three different organisms. And out of this merging of all these datasets, one big dataset was created, and then other two datasets were obtained by filtering out some proteins such that the similarity between the protein sequences in the dataset is less than a specified threshold. So with two chosen thresholds, we can get another two datasets. In this is something that I haven't mentioned. So the positive interactions can be obtained experimentally. That is clear. But how to choose the negative interactions? We can have, so they were chosen so as to have pairs that are not known to interact. But not all of the possible pairs that are not known to interact have been included in the negative set. But these specific data sets were created to have a approximately balanced number of positive and negative interactions. And this table shows the results that we have obtained on the four data sets and on each of the three architectures. So one denotes the baseline early fusion architecture, while 2 and 3 are the Siamese ones. And we can see by looking at the table that on all data sets, the best result is obtained by one of the Siamese architectures, thus suggesting that having such an architecture can improve the performance and better model the relationships between the two proteins in the pair. Further, we have compared our approach with other methods that report results on these data sets. And on the human data set, we can see that our approach outperforms the majority of related work with three notable exceptions. The best method being an autoencoder on graphs, so the one that I talked about earlier. And on the multi-species data set, we were able to obtain slightly better results than the other approach that reports results on this data set. So even though the improvement is not very big, it is consistent across the three data sets. To conclude, in this presentation, I have presented a procedure for binary classification of protein-protein interactions, in which we proposed two new Siamese architectures for the autoencoders, and we showed that they can improve the performance on the four evaluated datasets. These datasets included were varied in the sense that they included proteins from different species, and our approach was able to surpass the majority of related work on these datasets. When looking at future directions of research, we can think that the results that I have presented earlier, both of our method and from the related work, show quite good performance, so above 90% accuracy, let's say. However, this method of evaluation in which we randomly sample pairs does not show the full picture, in the sense that it does not take into consideration whether the testing proteins, one or two of the proteins in the testing proteins pairs have been seen in the training set in combination with other proteins. So this aspect proves to be a relevant one because multiple works, including the ones that I've listed here on the slide, have observed that there is a drop in performance when testing on unseen proteins. So if we think of the protein interactome as a graph. This would correspond to being able to detect edges for a new node in the graph that is not connected to any other nodes, as compared to being able to detect missing edges from nodes that are already contained in the graph. An important future direction of research is being able to improve the generalization of such prediction methods on unseen proteins. Another aspect that was not addressed in this work is that of dealing with imbalanced datasets, because as I mentioned, the datasets that we've been working on have been built such that the two classes are relatively balanced, but this does not correspond to a real world scenario in which the actual data is highly imbalanced. Lastly, a very important aspect is being able to provide interpretability for our methods, for example, by being able to identify which of the amino acids are actually involved in the interaction. This would conclude my presentation. Thank you.